Hello and welcome to Nikolai's genetics lessons, and today's question is: if there are 40 centromeres in a cell at anaphase of mitosis, how many chromosomes will be found in each daughter cell following cytokinesis? In order to answer this question, let me use this picture, and on this picture you see the cycle of the cell. The interphase would be the longest phase in this life cycle of the cell, and how chromosomes would look like. They would be relaxed and would look something like this. And what's going to happen during this phase? DNA should double. So if we say that this is how chromosome looks like, and at the end of this phase, the chromosomes would double, and this is how chromosome would look like. We still call this chromosome, those now it consists of two sister chromatids. But starting with prophase, chromosome would condense and would look something like this. So it's also very schematically and would look like this. Just like on this picture, we see two sister chromatids that makes one chromosome. Now we also see chromosome that consists of two sister chromatids, so these two sister chromatids genetically identical and result of the self-replication due to semi-conservative nature of the DNA would be identical. How many centromeres we would see? We would see one centromere here and one centromere here per two sister chromatids. During metaphase, spindle fibers would attach to the centromere. So let me show it here. So would attach to the centromere. It's not necessarily have to be just one. It can be many, hundreds of them, and would pull these two sister chromatids into different directions and would break this chromosome. Again, we call this chromosome. And what's going to happen? Next stage, we are going to see two sister chromatids that separate. So one sister chromatid, another sister chromatid separate, and now we would see two centromeres. And now we call this not sister chromatid but chromosome. And this we also do not call sister chromatid anymore but call chromosome. So instead of one chromosome here, now we have two chromosomes here. Instead of one centromere here, we see two centromeres here. And this is exactly what we see, what happens during anaphase. One more time, during anaphase, chromatids of each chromosomes separate into daughter chromosomes and move to the opposite poles. Number of chromosomes double and number of centromeres would double. So I show centromeres here, 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 and here. And if during this stage we can count 40 centromeres here, according to our problem, 40 centromeres during anaphase of mitosis, that actually means that 20 would go in one direction to one pole and 20 would go to the other pole. Using this picture, so 20 here, 20 here, 20 here, 20 here. And as a result, 20 in one daughter cell and 20 in another daughter cell. Let's read our question again. If there are 40 centromeres in a cell at the anaphase of mitosis, how many chromosomes will be found in each daughter cell following cytokinesis? And the answer is going to be 20. And by the way, how many centromeres we can find in the metaphase? I hope now you know that we can find 20. 20 here and 20 here. So don't be confused. 20 in interphase, 20 in prophase, 20 in metaphase, 40 in anaphase, 40 in telophase, and 20 in daughter cells. And this is all for today. Subscribe and see you in the next video. Goodbye.